go ahead and get started. Welcome everyone to Plants to Plants, the hemp and cannabis processing seminars. I'm Emmett McGregor, CEO of SciPhy Systems and your host. Today we are joined by Trevor Murray of Zaiput Flow Technologies, one of our most exciting new vendor partners in the space of liquid-liquid separations, liquid-liquid extractions. We've been doing some amazing development work in partnership with them, and we're just thrilled to be able to dig into some of the capabilities of Zaiput Technologies today. Trevor, would you like to introduce yourself?
Absolutely. Well, thanks for having me on, Emmett. I think this is really exciting and great to get the word out about Zaiput's technology and show the folks what the power liquid-liquid extraction can do in your cannabis processing. But to get started, my name is Trevor Murray. I'm the Sales and Applications Director over at Zaiput Flow Technologies. I've been with the company for about 4 years. My background is in chemical engineering, but I spend a lot more of my time today working with people on their problem solving than, I guess, true chemical engineering. And I've been, as I mentioned, with Zaiput for about 4 years now, and we've seen some tremendous growth with the company over the years.
A lot of growth even just since we've been talking. So a very exciting time for both of our companies. And can you tell me a little bit about the origin story of Zaiput Flow Technologies, where you came from, how the technology was first developed?
Yeah, absolutely. So the technology got started at MIT around 8 years ago. Our founder and still current CEO, Andrea Adamo, invented the technology there when he was in Klaus Jensen's lab. It's a lab really focused on flow chemistry and making continuous processing work. Their original idea was to try to make something about the size of a refrigerator that could make every drug possible. So maybe if you were going to go onto the battlefield and a soldier needed a certain drug, you could use this refrigerator size thing to make any drug possible. Of course, the reality fell a little bit short, but one of the shining gems out of this collaboration and idea was our core technology, which is our liquid-liquid separator. Since that time, there was such interest in the technology that our founders spun the company out of MIT. And over the last 8 years, he's been working on building Zaiput into what it is today. So this includes scaling up the technology, building much more complex technology. And I think probably, as we will discuss a little bit later today, is different offshoots of where this technology can be used. So over this time, Zaiput has grown significantly, really all funded by our own sales and interest in the technology. So it's been quite a fun time for the company. One other fun note that I guess I'll mention in the history of Zaiput is in 2018, with the opportunity to launch one of our devices into space. Maybe as we'll discuss a little bit later, we have a completely gravity independent separation, and we wanted to prove that in a place where there's really no gravity. And so we launched a device up to the International Space Station in 2018, had some astronauts run experiments with the technology and found out that the separation works just as well in space as it does down here on Earth. So when cannabis production makes it to Mars, I think will be a great solution for separations and extractions there.
Amazing, yeah, and I can imagine there's a lot of very tight liquid streams, both waste and process streams up there at the space station where this could be applied. So I'm sure you got some interest there. Speaking of, working with NASA and the ISS, but who else are your main clients, outside of the cannabis space? What can you tell us about the sort of first applications that you went after?
Yeah, it's a really good question. So we started basically since this first collaboration with MIT was with Novartis, a large pharmaceutical company, and they were the ones really trying to push this refrigerator-sized machine at first. That is because of that, we really ended up working primarily with the pharmaceutical industry, especially early on in our company's history. They're still one of our largest clients, if not the largest client. We work with 19 of the 20 largest pharmaceutical companies in the world, and have now taken a real focus onto the manufacturers for those organizations. So these are CMOs and CDMOs who are the ones actually making the drugs for these pharmaceutical companies. And a lot of them have found that our technology can really help them improve their processing. Outside of those types of folks, we do work with the National Lab System here in the US, a lot of other prestigious research organizations around the world, including some of the top end universities, of course, our alma mater, MIT, but other universities, you name it, UCLA, Purdue, USC, universities across the country and across the world, we see, including specifically Europe, are using our separators as well. I guess the last little bit would be other industries like flavors and fragrances, fine and specialty chemicals, and now, of course, with you guys, the cannabinoid and hemp industries, which is kind of our new area of growth and something we're really excited to work within.
Great. Yeah, and so the problem really that you're trying to solve and that you're effectively solving for these businesses is, liquid-liquid separation, so separation of two liquid streams, or also, I understand, breaking emulsions. So the biggest interest that we're seeing in our space typically is a liquid-liquid extraction process using two solvent systems to achieve some form of separation. Can you explain a little bit about what that liquid-liquid extraction process really is and how that applies to our space?
Yeah, absolutely. So liquid-to-liquid extraction is a really common processing step, especially within the pharmaceutical industry. It's one of, I would say, the oldest processing steps. It's not anything new and fancy like we could imagine with chromatography or CPC or something like that. It's been around for a long time, and there just hasn't been a lot of innovation, I think, around it until, of course, Zaiput came about. But in a really simple sense, liquid-liquid extraction is moving some sort of solute from one phase to another. And this is typically in organic aqueous pair that's going to form two separate layers in a gravity separation type methodology, right? And so the ideal scenario would be you find one phase that's going to selectively capture either your product or all of the impurities except for your product, and selectively remove them from the other phase. So you end up with then a product-rich phase and an impurity-rich phase.
Right. And when we're talking about phases, in this case, we're really talking about two liquid solvent systems that will separate from one another under the right time duration in a gravity based separation system. So oil and water being the most commonly understood version of that.
Yeah, that's a great clarification. Some folks will come to us and be like, can you separate water and methanol? And unfortunately, those aren't separate phases, right? They're one cohesive phase. So we'd have to separate like a water methanol phase from an oil phase or something to that effect.
Right. So I think a couple of examples of processes that we've seen in the industry using this already are things like brine washes of alkane solutions, say pentane or hexane, where that brine can then be separated, as an aqueous phase apart from the alkane solvent, which is, an organic phase. And typically that's just done putting it in a mixing tank, letting it settle over time using gravity, which can take many hours. So let's talk a little bit about other types of processes that use liquid extraction, or like what is the actual function or process that's happening in that mixing and then settling type stage that would be the alternative to what Zaiput can do very quickly, efficiently, and continuously.
Yeah, definitely. So the most traditional method of doing this extractive step is, as you've described, it's putting your product phase and your solvent phase, or your product phase and your wash phase, whatever those may be, into a giant tank and mixing for a period of time, 30 minutes to an hour usually, and then letting that gravity separate out over time. For processors who are maybe less familiar with this process step, you can almost think about it like the salad dressing in your fridge. You shake that up and it emulsifies, and then over time it gravity separates back out. Same concept. Now, this is a very traditional way of doing it. Inherently, there's nothing wrong with this process step until you actually get into trying to bring this into a productive factory type environment, right? What can really cause challenges here is how long that takes to gravity separate out, particularly with natural products. We can see this being separations that can take hours to even days to gravity separate out. And now if you have even, say, 1,000-liter reactor, you're putting that on hold for multiple days for this single process step. It's awfully inefficient, right? There's lots of advantages to liquid extraction, large capacity, low energy requirements, but you lose some of these advantages when your productivity goes from, 1,000-liter reactor maybe in the course of hours to a 1,000-liter reactor in the course of days. Now, there's other methods of doing this. You could imagine centrifuges are a very common method of doing these types of separations. They've been around for a while, and I haven't seen them broadly adopted, which can kind of tell us a lot about why they are not being used. There's can be issues from challenging separations even with them. They're not working as effectively as you want them to long optimization times, and then replacement parts and cleaning can be a big pain with centrifuges. They're obviously beautiful pieces of equipment, really high tech, but with that comes the ability to break really easily, and it's challenging to repair. So
yeah, huge forces. Just so many G's of force on those bearings when you're trying to rotate a piece of equipment that quickly, and inevitable breakdowns happen so fast, but also moving very fast. Often they have safety precautions built in, but that's a lot of mass moving very quickly, a lot of force to be handling. So moving away from that reliance on very fast moving equipment to something that's more of a membrane-based approach that's reducing the number of moving parts and increasing reliability. That was something that was very appealing to us and to our customers. Can you tell us a little bit about the actual liquid extraction function, though? We have these two phases. We have something mixed in there that we're trying to separate, but how is that actually functioning? What are the forces at play, and how does that affect the process design?
Yeah, definitely. So a couple different things here that I guess we can dive into. The first is the first step, I guess, in an extraction is really mass transfer, right? We want to move compounds from one phase to the other. And in order to do that, you have to promote a lot of mass transfer, right? So often in in traditional systems, this is done just by mixing in batch. When we bring that into our context, we do what we call inline mixing. So basically you tea two phases together, you do a bunch of mixing, and then we run through a phase separation. So of course, in batch, that phase separation is driven by gravity and time. That's going to be a density difference between your two phases. There's a lot of other forces at play. But for us, we use a membrane-based technology that can pretty readily separate these two phases rather quickly. And so it's a completely flow through type device. And I can actually bring up a quick diagram of how this works, if that's all right with you.
Sure.
Great. So it's a membrane-driven technology. Rather simple, as you can see. There's only about two moving parts and two functional parts within this whole device. Essentially, we have a membrane that's going to be what we call is selectively wet by one of the two phases. And that's a different term, I think, than what a lot of people think when they think of membranes. Most people will think of, say, a tangential flow filtration membrane, which is really size exclusion based. Our technology here works with wetting properties. So you can imagine what happens is when a droplet of the phase that is supposed to wet the membrane makes contact with the membrane, it'll actually spread out and coat it and soak into the membrane, while the other phase is going to be repelled. The best example I could maybe give of this is if you have a frying pan in your kitchen. If you take oil and drop it on that frying pan, it'll spread out, coat that pan. But if you took a drop of water and put it on that frying pan, it would roll around kind of, as a drop of water. It's going to be the same concept with our membranes. But now when it's a membrane, that wetting phase can actually pass through, and the non-wetting phase is repelled. So in this really simple manner, all we do is flow a biphasic stream into the device, and the membrane then allows for separation. The other little key aspect of this is a little pressure controller in the device that provides enough pressure to push that wetting phase through, but not so much pressure that we push both phases through.
And I see a question from the field about, okay, this is a great process. How does it apply to cannabis? So there's many different applications we could see here. On the non-wetting side versus the wetting side, it's going to depend on your solvent system, which of those is carrying which compounds. But For example, you could be talking about pulling pesticides away
Mm,
from your cannabinoids. And that, say, the pesticides in theory could go with the organic phase, and the cannabinoids could go with the aqueous phase, which may be, things like alcohols and water, or, any number of other solvent systems that are, essentially polar type solutions, right? Another process that we've had great success with is separating cannabinoids from one another.
mhm.
Another process would be purifying the cannabinoid stream of colorant type bodies, and um, any sort of separation where there's a difference in partitioning coefficient is possible using this liquid-liquid extraction stage. So we just looked at, how we're actually separating those two phases from each other, which is the unique part of Zaiput, where you're doing that, continuously in a very small footprint, and you can do your mixing continuously as well. So you're very much intensifying the process, reducing the amount of solvent holding capacity needed for that. But if we look at the liquid-liquid extraction process itself, what can you tell us about what's driving the process development behind that, and how it can be applied to this cannabis space for these many different applications?
Absolutely. Yeah, the real key of this is, it's real cute to separate two phases, and even if you have a nice membrane technology about it, so what? Why does that matter? Well, the real goal of this is to be able to selectively capture impurities in your process, and those impurities could be different depending on, what type of processor you are. As you mentioned, this could be trying to remove different pesticides, it could be trying to do terpene removal, it could be trying to remove colorant, it could even be separating different cannabinoids from each other, right? We've taken a look at a few of these different cases and seen some success there already, and that's something that we can maybe dive into. But the real key is, if you find the correct solvent system, then you're actually able to, at high efficiencies, remove whatever you define as your impurity. You can take that out of the process, or, kind of vice versa. If you have a single compound that you identify as your product, there's also the opportunity to selectively remove that single compound from the rest of your processing solution. So this can be a quick way to go about purifying your process stream, or your batches of material. Now, most folks, I think, today commonly will do this with chromatographic type methods, PrEP, HPLC, countercurrent partition chromatography. And those can work all right for certain compounds, but as I understand it, and you can maybe even say a little bit more to this, is there's some inefficiencies that go along with those, maybe some capacity problems as well, that can be really solved with our technology.
Absolutely. Yeah, what we see from the other approaches to doing things like cannabinoid separations, or even this pesticide remediation, which is always in high demand, especially the hemp side, but increasingly also in the cannabis side as we get more broad acre plantation type states. And that is, you know, the advantage of the Zaiput system for this type of application is that in something like chromatography, you have a solid phase that you're using that is either very moderately expensive and replaced frequently, but that limits your capacity based on the volume of that solid phase that you can hold. And you're talking about typically quite high pressure vessels required for that, so expensive hardware up front, and then a relatively high operating cost as you have to replace that media, or take a lot of time, on the other hand, to regenerate that media using, if it is a more like a polymer type media where you can regenerate its capacity. That takes a lot of time. The media itself is quite expensive. So using a liquid-liquid process rather than a chromatography process that uses a solid phase, you're eliminating some of the most costly operating expenses out of that process. When you look at something like countercurrent liquid partitioning chromatography, which is sort of the most similar process to what we're talking about that's currently deployed in the cannabis industry outside of Zaiput, the issue there is that yes, you're eliminating the solid phase, you're using two liquid solvent streams and then mixing and separating them. However, because of the centrifuge, you have a very costly upfront moving heavy mechanism that you have to deal with, and they have really bad reliability issues. But not only that, you typically don't see as much control over your mass transfer phase. You have essentially a fixed architecture of your mixing sections and your separation sections. So you're essentially having to upfront configure your hardware and be locked in to that hardware configuration moving forward. Whereas with Zaiput, we see a lot of flexibility. It is completely modular, individual separator sections that can then be tied in together to form a similar countercurrent mixing and then separating type configuration. So much more flexibility, better reliability, eliminating the solid phase, going to only liquid solvents for these separations, but then also the ability to dynamically adjust the hardware on a modular basis as you go. And typically, also from our experience, reducing the capital expenditure quite substantially per unit capacity as compared to a centrifuge, which by its nature just has high precision parts like bearings that are necessary for that type of system. So that's my experience. And actually, this is probably a good time to start talking a little bit about that countercurrent liquid-liquid extraction process. We've talked up to this point about a single stage, mixing two phases, extracting one compound out of the other solvent phase using one of the solvents. So, for example, we might say that you have a solution containing CBD with THC in it, and you're trying to get the THC out of that mixture. The way you're going to achieve that is by removing a portion of the THC using a fresh second solvent system, and then mixing them together, separating those two solvent streams, those two phases. But sometimes one stage isn't enough. So how do we go to a multi-stage system, and how do we gain resolution to really be able to get into those fine separations and achieve really efficient separations and go down to very low levels of our contaminant?
Yeah, absolutely. It's a great question, and it's particularly relevant for the cannabinoid space, just because so many of these compounds are so similar to each other, of course, right? So, within liquid-liquid extraction, we're really leveraging differences in solubility between the compounds. And if compounds are, awfully similar to each other, say take CBD and THC, awfully similar compounds, in the grand scheme of things, there's differences in solubility between, say, a hexane phase and a water phase may not be significantly different, right? So we have to, if there's only small differences, we have to repeat this extraction over and over and over to get to the levels of purity that we'd like to see for any given product, right? And you can do this in a very effective way with our technology by bringing it into a countercurrent extraction type setup, like we have up on the screen right now. So simply one feed material is going to be flowing in one direction, and the other solvent or wash is going to be flowing in the opposite direction. And because of the countercurrent nature of this, you can get much higher recoveries at much lower solvent costs than, or solvent amounts, excuse me, than if you were, say, doing this in a traditional batch manner. And so with our technology, you really have the flexibility to add as few or as many stages as you'd like to get to the levels of purity that you need. And this can really be determined pretty well with single stage extractive experiments that we can then extrapolate out and say, okay, based on what you see in one stage, here's what you could imagine seeing on 5 stages, 10 stages, or 20 stages with our technology. And obviously your resolution is going to improve with each additional stage. Now, how many you need is going to be specific to your process, but the beauty of the scalability of this can be that we can find pretty readily at small scales how effective that extraction or separation is going to be, and then take that and bring that all the way to production for folks.
Right. Because this process is based on this relative solubility in the two solvent systems that we're using, that doesn't change relative to the volumes involved, so long as the two solvent streams are mixed effectively together. So you may see changes in the mixing section. We're adding things like static mixers and things like that to increase mass transfer, increase the contacting and mixing of the two solvent systems together. But functionally, the separation stage, which is where the Zaiput separators really shine, doesn't change. It's essentially the separations based throughput capacity is relative to the surface area of the membrane effectively. So for that reason, small benchtop type of tests directly translate into production throughputs. So that allows for very efficient process development in the lab. So can you tell us a little bit about, I'm a client, I'm coming in here, I have a cannabinoid separation, for example, that I want to perform, maybe trying to separate two isomers of THC, for example. What does the process look like for me to get engaged and to get that process from concept stage through to production?
Yeah, it's a great question. And it's actually going to take us on a little bit of a detour away from what we've been maybe already talking about and towards a new bit of technology that Zaiput's releasing towards the market. And that we're really actually quite excited about, because it's going to answer this question that I think we got earlier on that was like, okay, great, so what, right? You have this liquid extraction capability, you have this technology. What's that actually going to do for the cannabinoid industry? Because in the end of the day, all is going to come down to is how well can you separate specific compounds from each other? And that's going to really be a function of your solvent system, right? It's going to be a choice of, am I using, say, toluene for this process? Am I using hexane for this process? Or am I using ethanol for this process? And that can be a difficult question to answer for your average chemist, right? Or average cannabinoid processor, because maybe one, you only have a couple cannabinoid or a couple solvents on hand. Two, you only have so much time to look into this. And three, you maybe wouldn't even know where to start, even if you had infinite solvents and an infinite amount of time, right? So what we've done at Zaiput is built a high throughput solvent selection platform. And so the idea is a customer would come to us and give us a parameter that, let's say, for example, they wanted to separate CBD from THC. And they would say, my parameter is I want to do that. I want to keep the yield of CBD as high as possible, and I want to remediate THC to below 0.3% or something to that effect, whatever their parameters are. We can take that and plug this into our high throughput experimentation platform. And then what the platform will do is it's going to take a library of solvents that we have on hand, and it's going to run through various extraction experiments on our starting material. So essentially, a customer would take their exact material that they're using in their processing, send us a small sample of it, right? We're talking tens of milliliters, so really small volume. And we would then take and run this screening on it and find the exact system that's going to work for that specific customer. We come back to them with the data and say, here is what you could be looking at if you were to work with us eventually, right? At a single stage, at 5 stages, at 10 stages, right? Here are the different extraction efficiencies you could imagine. Here are going to be the recoveries of your product, the losses of your product, right? And then, of course, we can do further development work ourselves. This is then up to the customer whether they want to have Zaiput do some of the development or if they would like to take the development project on for themselves. It's personal preference there. And pretty readily, we can generate the data necessary to have a high degree of confidence to go to production type environments with our technology. As you mentioned earlier, the scalability is quite readily available. All you have to do with it sounds simple on paper. In reality, it's a little bit more challenging to build the larger separators, but all you have to do is make the membrane area larger. And then we've written an extremely boring white paper about mass transfer that I won't dive into because I'll put half of the audience to sleep. But we can fairly readily scale this process off of these types of lab results. So that's a long-winded answer, I think, to that question. But I think there's a lot of different aspects of coming in and working with us that we can really assist a customer with.
Absolutely. I mean, it's shouldn't be understated the value of that automated solvent
Mm,
system selection platform and that being offered as a service to clients upfront before purchasing large scale equipment and investing a lot of money into hardware. You have the opportunity to use Zaiput's expertise and send actual samples into their lab and have them run experiments that if you were trying to do it in your own facility using conventional methods would take potentially weeks or even months of work and consolidate that into a very short turnaround time so that you can start from day one with a solvent system that's not only verified for the process, but it's verified using your material and has essentially multiple pathways, multiple options to best suit the outcomes that you're targeting using your material. And this is something we've heard many times from the field from folks using, countercurrent fluid chromatography or even other types of chromatography systems out there that they simply did not get the process implementation and development support that they needed to achieve the outcomes that they were looking for within a reasonable timeline. So it's really our focus to say, up front, let's validate the process, let's find the right process solution custom to your application, and then go from there in building out the hardware system that's going to be optimized for that process. And we've seen great success. We've seen some isomer cannabinoid isomer type separations. I've seen some questions about that in the Q&A there. Yes, we've seen successful results on those types of separations. We can work with you to achieve those types of separations. Pesticides and heavy metals type separations, those are going to be typically easier than separating very close, very similar compounds like cannabinoid isomers. So that's something that would be straightforward to get a lab test done and to then implement that process at your facility. So we work hand in hand to validate the process up front with the actual material that our clients are going to be using so that there's no question that it's going to be effective once it comes into their facility. And again, if you prefer to have some degree of in-house expertise doing that type of testing after the initial consult, that's completely acceptable. We do have hardware lease programs available for that, as well as purchase available for the smaller benchtop and then sort of intermediate stage throughput production type separators that you could use with your staff and your facility to, again, further validate, further develop, and optimize those processes for your particular product. Something I think it's really important to understand about this discussion, though, is that much like more advanced chromatography solutions out there in the pharmaceutical and fine chemical space, uh, we're not taking a one size fits all type approach here, right? This is a technique which requires to achieve high efficiencies, to reduce the amount of loss, to maximize the effectiveness of the separation. And when we say loss here, I want to be clear, we're not talking about destructive loss. What we're saying is that the fractions, the two fractions that are output from the process, the fraction containing the components that you're not looking at as your product will have a higher percentage of the product compound. So it's not destroyed, it only goes into a separate solution stream, so it can potentially be recovered for you. But taking a step back, again, when you go to chromatography, there are sort of generalist type resins or types of medias that are sometimes used, but to achieve the most precise type of separation, you're going to do experiments to find the right media for your solution, for your process. Same here, we're finding the exact balance of solvents, not only identifying the solvents, but finding the right percentage and proportion of those solvents to one another to achieve the exact separation for your specific process. So each process may need a different solvent system, a different proportion of solvents, in addition, different actual solvent compounds to achieve the optimal results. This consult service with Zaiput allows you to identify those solvents and their proportions relative to one another very quickly in a way that you're not going to be able to do at your own facility without huge investment in analytical capacity and process development type hardware. So a huge service being offered here. And that said, the hardware itself, the separators, the mixers, the pumps, everything that goes into that implemented hardware solution is cross-applicable across multiple processes and can be effectively used for multiple types of process outcomes. So,
yeah, I think that's a really nice point, actually. The concept of, if you have, say, multiple processes, like one maybe where you're trying to separate different different isomers of THC from each other, maybe another one where you are trying to separate CBD from THC, and then a third process perhaps where you're trying to separate terpenes from each other's, or terpenes from your cannabinoids, right? You could be running all three of those processes in your plant, all on one piece of equipment that Zaiput supplies. Now, you're going to need a different solvent system to run each of these different separations. But with this quick upfront development work that we've discussed, you can find what those systems are, and then you could almost run those processes sequentially on the same equipment, or of course very readily even add more equipment and run it all in one line. But that is up to the user's choice. But I think it's a really good point that the equipment itself is very versatile for all of your processing. It's just the solvent system that has to be specific to your specific process.
absolutely. Well, let's see. Want to make sure we're covering all of the questions in the field there. Please, anybody, if you have additional questions, use the Q&A section here. We have an anonymous attendee who's submitting a lot of questions, which is great, and I hope we're covering all of those for you, but we do want to hear from anybody else out there in the field as well that may have additional questions or points of clarification they'd like to be attended to in this call. As far as operating cost differences, there is no blanket statement we can make there. In different processes call for different solvents cost different amounts of money, different amounts of loading capacity for different processes. So depending on your specific process, the cost efficiency is going to be different. However, in a lot of cases, liquid solvent systems are going to be more cost effective than liquid and solid phase types of chromatography systems for a lot of processes, especially because in a lot of these chromatography processes calling for very fine separations, you're having to use very high dilutions of solvents and also pay for relatively expensive solid medias. And so we can oftentimes reduce the total solvent volumes being used for a given process, and also those solvents can be renewed in many cases to be recycled and reused in your process again. And the same thing does apply between solid phase chromatography and this liquid extraction, where solvent recovery is required to then repurify the solvent, and that applies to both. So we consider that to be an equal requirement. But the solvent volumes utilized may, in many cases, be substantially lower, and there is no solid media to be replacing, which is often quite expensive. Also, the hardware capex upfront can be substantially lower depending on the specifics of your application.
Absolutely. Yeah. I think the note of a lot of the work that we've done already has been really at the solubility limits of what we can do with these compounds. So we're really trying to push the maximum solubility which is key for processors, right? If we're running this as dilute as you would have to do on a chromatography setup, there may not be as much advantage, right? But we really want to push the limits of how concentrated we can get this for maximum productivity in the plant. And along the note of productivity, I guess we haven't really talked about how large and how small the technology can get. In a basic sense, it starts right in the lab. It can sit in the palm of your hand at our smallest sort of devices, which you would see in the Sep 10 is really the size of the palm of your hand. And then we scale all the way through a few orders of magnitude up to our current largest device, which we call our SEP 3000. That processes 3 liters in a minute or 180 liters in an hour. And we're seeing this around about 10% dilution. So I guess this would be about 18 liters of your starting oil per hour. Soon, we're going to be scaling up even further to a 40 liters per hour unit, which we are quite excited about. That should be coming out in the next couple months here. Sorry, that's 40 liters per minute unit. So 2,400 liters per hour. So you could be processing, I guess, of liquid volume, of oil volumes, that would be somewhere in the realm of 240 liters per hour. So some serious throughputs.
Right, and just to further clarify that, the throughput is rated at the feed volume input, so that includes both of your 2 solvent phases and your solute oil, which is going to be separated. That said, very high loading capacity, depends on your process, can be quite concentrated, and so see very good throughput. And the value that we've seen for processes like THC remediation from CBD, like some other processes that are being tested, targets like pesticide remediation, terpene separation from cannabinoid oils. All of these things have the potential to have very good capex as compared to other available solutions. So there's good economy of scale as you go up into the SEP 3000 stage. You're going to see total hardware packages that are coming in well below comparable, prep HPLC type equipment or things like that that are commonly used for this process. So please do talk to us at SciPhy Systems. Our salespeople can walk you through your specific process. We'll get you on the call with Trevor to walk through the nitty-gritty specifics of the chemistry requirements and the hardware selection, and move forward as quickly as possible towards a consulting type arrangement where we're testing your actual samples and getting a process optimization done for you. So I encourage everybody, again, please do submit additional questions. I love seeing some others joining in here. Let's see. There's a question about the membranes retaining pesticides or contaminants across multiple runs. Can you tell us about your experience with that?
Yeah, it's a really good question, and it's actually something that we do get all the time. When people think about membranes, we really commonly think about these types of size exclusion type membranes that are going to maybe even be activated and have ligands on them that are going to be maybe selectively picking out materials from our process stream. And that's simply not the case with our membranes. We really look to have this really large pore size membranes. We're looking in the range of, hundreds of nanometers to up to a micron in pore size, which may seem really small to you and me, but in the world of membranes, that is quite large. And so what this really allows for is it kind of keeps us at miscibility conditions, right? So we're not changing the miscibility. Nothing's going to be dropping out and getting stuck in the membranes, or we're not going to be changing, what can pass through the membrane essentially, right? So even these large pesticide type molecules are going to be able to pass right through the membrane with no problem if they are part of the phase that is going through the membrane.
Right. And one thing to really emphasize here is that some people may be familiar with the X-spiral membranes, tangential flow type membrane systems that we use for solvent recovery in ethanol and alkane type applications, and for dewaxing and decoloring in those same solvents. It's important to understand that we're talking about probably maybe a couple orders of magnitude difference in surface area as well. This is a very compact, small amount of surface area required for high throughput separations due to that pore size and due to the different mechanism of separation that's being used. So there's not really that much surface on the membrane to even retain compounds. Plus, the materials being used are very inert. They're specifically designed not to hold any compounds between runs. You will flush the system, just as you need to do clean in place for any type of process when switching between batches, but it's a simple solvent-based flushing type process. Yeah, these are designed for pharmaceutical applications where there are very, very low percentage compounds that could be considered toxic and therefore invalidate an entire pharmaceutical batch. So you can rest assured that a lot of attention has been paid to the cleanability and sanitary standard of this process equipment.
Yeah, I think
so
that's a good point on the pharmaceutical industry, right? These have to be FDA validated processes, and they set a really high bar. And so we have to work with our customers to make sure that we can really clean these devices and clean them well, because there's not margin for error in that type of world. So it's something that if you do have an issue, we'll work with you to develop a cleaning procedure that's going to take away that concern.
Absolutely. And one other thing, while I mentioned the comparison between X-spiral and Zaiput type membranes, as Trevor mentioned, the tangential flow size exclusion type membranes that are represented in the X-spiral product line, that is using pressure to force a single solution across a membrane to then hold back compounds of a given molecular size. That is a single solvent stream type process, and it requires high pressure and very high surface area. So you're seeing very tightly wound, spiral wound type of filtration modules typically. Now, in this case, what you're seeing with Zaiput is a wicking type wetting property based separation, where the separation is not based on pressure forcing the solvent through the membrane, but instead is based on the wetting properties of the surface of the membrane, causing one of the solvents to wick through those pores and into a separate outlet. The pressures are relatively moderate. This is not, in the 1,000 PSI range, or even up in the high hundreds PSI range. And the membranes themselves are easily replaceable sheet type membranes that are relatively inexpensive. Again, small surface area, small amount of material. The material itself is fairly specialized and is available exclusively from Zaiput. But that said, the cost of that is very low. So as a routine maintenance, you will replace it, but it's not going to be a major cost of operations, and it's quite a simple procedure. So
yeah,
let's see here.
To give an idea, as maybe as you read through some of these questions, is the lab scale membrane size is about the size of my pinky, and it costs about $4. So it's not something that we are really trying to rip you off. We're really advantaged by our ability to use convective flow and these small membrane areas. If we were working on the scales of large, say like RO type membranes or TFF type membranes, where you really do need large surface areas, it would be a completely different scenario. But because of our small pore sizes, we're really advantaged there. Or our large pore sizes and small area, we're quite advantaged there.
Great. Yeah. So one other question here from Tanya, and that's on alkaloid isolation from kratom and mushrooms. Now, we can say that SciPhy Systems has a great interest in testing that. We have not done that ourselves, but can you speak generally about alkaloid separations and purifications of those types of compounds? I'm sure that's something you've dealt with in the pharmaceutical space.
Yeah, sure. So these are types of separations that we do see all the time. It does always come down to solvent selection, finding the right solvent to selectively pull out the right alkaloids that you're trying to identify from your broader product mixture. Alkaloids can end up being a bit more polar than other molecules, so sometimes a simple water wash can do this quite effectively, and that could also require maybe the addition of salts or something to that effect that would selectively drive out some of these other compounds. But that's all things that our solvent selection platform can really help you identify. So to the specific end of, do I have a specific answer? No, I do not. But it's once again a separation question and challenge that I think could readily be answered with our technology.
Right, and something else to talk about here is that typically this process configuration we're using for most of these processes is either a single separator stage where you're creating two output fractions that are in two separate solvent streams, Or in the case of the countercurrent liquid-liquid extraction platform, you're having those same two fractions running counter to.
Yep. Hey, Emmett, I think we might have lost your audio for a sec there.
The microphone went out on me. Can you hear me now?
You're back. Welcome back.
All right, great. So we're going to take a step back then. Essentially, you typically have two fraction output
Yep.
streams. So you're making essentially one cut separation between the spectrum of different compounds you've got. If you need to purify a single isolated compound out of a broad natural extract type of spectrum of compounds, it's possible that the optimal solution may be essentially two process stages. First, separating compounds of, say, less polarity than the target compound, and then taking that output fraction containing the target compound, and then doing another process where you're removing the more polar compounds, for example. And that's just a rough example, but the specifics might vary more. It's not just polarity that's having an effect, right? There's many other forces at play here, but that enables a very, very pure output stream from a very broad spectrum extract. So that is a feasible type of process to approach with this type of equipment.
Absolutely. Yeah, you could imagine basically your chromatogram as it would come out of, say, your HPLC or something like that. There's theories of relative solubility that say you could basically take the left part of whatever's left of the peak and whatever's right of the peak, obviously the more polar and less polar components, and then leave a single peak in the middle. What we've also found, though, is solubility, even though it's been studied for hundreds of years, at least the last century, is still rather nebulous, and there's not a single theoretical solution to this. And sometimes brute force has to be the right solution, which almost shouldn't be given where we are with technology today. But that has been the case, and it's proven out by the fact that we found some non-obvious solutions using this high throughput experimentation platform that you wouldn't really anticipate. And you look at the maybe specific combination of solvents and say, why does this work and this other combination does not work? And that's why our customers will really like this, is they're going to find success with their specific system. It's not that we're trying to take a solution that is off the wall and apply it to them, and it may or may not work. We're going to take something that's really specific, or an approach that's specific to them. So for the folks who are trying to isolate their alkaloids, definitely reach out, and we'd love to be in touch and figure out the solution for you.
And one thing to mention here is that in actually a lot of traditional types of purification and extraction processes, especially in things like mushrooms from traditional Chinese medicine, or even kratom, which is oftentimes made into a tea, you're effectively performing a liquid solid extraction step first, to then pull your compounds out. And then in conventional things like alembics or other types of extractions, there's often a multi-stage process done to refine these compounds, even going back hundreds of years, right? So what we're doing is just modernizing the process development approach to using the liquid-liquid stage after the liquid-solid extraction happens, to achieve that purification to a degree of reliability, repeatability, and precision that, of course, in traditional hundreds of years ago type approaches, they had no method for testing for the output product. But it's certainly not something that has not been done through many generations, because even things like the sort of three-stage extraction of mushrooms, you might be extracting with, ethanol and a hot water extraction, and then doing some sort of distillation type extraction process. That's very similar in some ways to what we're seeing here, where you may be, using the water to extract polar compounds, and then using, alcohol or another polar solvent to pull the more I'm sorry, I may have that reversed, but anyway, you're trying to extract both the polar compounds and the nonpolar compounds out sequentially from the solids in that case. In this
Mhm.
case, what we might suggest doing is extracting broadly the compounds into a solution to get it into a liquid solution, and then separating them, not extracting sequentially from the solid phase, but extracting them sequentially from the liquid phase of the extract, so that you actually have less mass to deal with when you're
Mm.
performing that extraction. So you can have higher throughputs in the separation stage because you're dealing with less volume in that separation stage. So certainly something with a lot of tradition coming up to this point, but now we're modernizing the process and getting more and more exact in the type of separations we can perform. Um, I did see an additional question here about analysis. Uh, let's see. So there's sort of two separate questions about analysis, and I think they sort of converge on the same point here. So one thing to understand is that the process development solvent selection platform
Mm.
is provided as a service,
Yep.
right? So you can send samples to have that done in a very high throughput, quick turnaround type of framework with Zaiput. But that is not a piece of hardware that is sold to customers at this point.
Not at this
Uh,
point.
right? So maybe off in the future, that's something that research and development type companies might be able to purchase to have in-house. But as it is right now, that's a service that is provided. But then taking those results and the lab results, which could include, multi-stage countercurrent liquid-liquid extraction at Zaiput's facility, that may take a little bit more time to put in the development queue, but it is something that can be made available as a service, or alternately doing that at your facility with equipment in your facility on the bench top scale or moderate production throughput type scale. Um, the way that that is then going to be tested is using the same sort of analytical methods that you would be using for compliance testing or for in-house Q&A type testing. You could rely on a third-party analytical lab or in-house analytical capacity to perform those tests to validate that your process is achieving the same outcomes as were in the original process development. What we've seen up to this point is that, again, very very easy scale up. It's almost always quite similar outcomes from the small scale up to production scale, much more so than a lot of the other processes that we typically deal with. Um, so I hope that answers some of your questions, but additional analytical capacity either from third-party labs or in-house HPLCs or GCs types of testing equipment where you have an in-house method for testing for the compounds that you're trying to either eliminate or to retain. As long as you have those methods on hand, you should be able to easily validate the results of the implementation of the larger scale equipment.
Yeah, maybe a couple notes to add on to that too. What we actually ask for very often is the HPLC or UPLC methods from our customers, so that we're comparing apples to apples with them. The peak that you're expecting to come out at 4 minutes is coming out at 4 minutes, regardless of what your starting solution is. And then, of course, if you want to do further analytics, we can ship you samples back. You can send them out to whatever labs that you like, or use your own in-house capabilities. And then along the notes of the scaling, the concept remains exactly the same. Of course, we've kind of gone over the scaling argument, but the outputs should be very similar to what we find at the lab scale. And if it's not, Zaiput is here to work with you to get it to the point where it is. It's not a case where if you get our equipment, scale up with us, and then start going into production and you find something that isn't working for you, that we say, forget about you, figure it out yourself. That's not the way that we work. That's not the way that we operate. We're going to be there to work with you through that until you find what we were looking at the lab scale. So I would say that on the analytical and the scale up methodology of the analytics, that's where we would be.
Yeah, so we are available to provide on-site and off-site support between SciPhy Systems and Zaiput Flow Technologies. We're here to see you all the way from initial testing, solvent selection, a benchtop scale up of the actual process, and then full-size production implementation. That's something we will walk hand in hand with you through that process and offer on-site commissioning and support services as necessary. So, we're aware that there have been other sort of horror stories, especially from the CCPC type of implementations where people have been sort of marooned without functioning processes. That's absolutely not how we work. It's also a big part of why we say we want to test your material from day one. We want this process to be specified to your actual output characteristics of your production material, so that when we go and we scale up, we know that that's actual transferable knowledge and not genericized tests from some other sample which may or may not match the characteristics of the actual material that you're using in your process. So that's very important to us, and it does mean that we go through with every client through a rigorous validation process to assure that your larger scale implementation is fully.
Might have lost you again there, Emmett, just at the very end.
We can give you that assurance of the process from day one before you heavily invest capital in the hardware. Or if you're on an accelerated timescale, of course, you can scale up the I apologize with the mic failures there. All that is to say, we're in your corner from day one, and we are making sure that you invest as little as possible up front to then apply to the large scale implementation, and that you order that equipment with confidence that your process is going to be effective. And we can work with different clients on different ways to make sure that you're meeting your implementation deadlines, but also have the information you need on your process to have effective implementation. So I think we're through all the questions here. We are running out of time with Trevor here. We might be able to take one or two more questions, but sneak them in now, or else we're just going to have to say goodbye. While we're waiting for that, I want to thank everybody for attending. We really appreciate you taking time out in your busy work week to come here with us and learn about Zaiput and how it might help your business. Again, I'm Emmett McGregor, CEO of SciPhy Systems. We provide equipment and processing solutions for the hemp and cannabis industry, as well as other natural products. And thank you so much, Trevor, for coming on and representing Zaiput Flow Technologies. Do you have any last words you want to give to our audience?
Yeah, well, thank you very much for having me, Emmett. I think this was a really nice time. I love talking about our technology. I think it's a great bit of kit for folks in a variety of industries, and I think it can definitely help out the hemp and cannabinoid industries as well as others. Any folks who I saw, there was one question who's coming from mushroom space. So I think that's fantastic. I would say one other thing is we really are a group of motivated and nerdy engineers who love to solve problems. So if you have other questions that are basically outside of maybe what we've covered here today, or other process challenges that you foresee for yourself or the industry, please do reach out to us. We love trying to solve these problems and would love to hear how we could potentially help you out. So definitely keep us in mind with any engineering problems that you may be looking at. That's what we do. That's what we're focused on. But thank you again for having me here. I think this was a blast today. I'm glad that we're able to get the word of liquid extraction in the hemp and cannabinoid space out there. And I appreciate the time.
Well, thank you for coming out, Trevor. And just to remind everyone, we have had successful tests in separating THC isomers, in separating THC from CBD, and some other cannabis-related processes. We're engaged in further research today around a variety of other separations. High priority applications are pesticide remediation, improvement of output oil quality, separation of terpenes from cannabinoids. There's a diversity of applications that we can help you with this technology, and we'd be glad to speak with you. You can reach out to info@sciphysystems.com. Find us at sciphysystems.com, at SciPhy Systems on Instagram, on LinkedIn, and Facebook. If you're coming to see this on YouTube, please like and subscribe for future episodes. We really appreciate you coming out and taking your time to view this episode with us, and stay posted for future episodes. We're going to have a cultivation and processing optimization episode upcoming next. with agro lighting. And thank you again to everyone for attending. And thank you, Trevor, for coming out.
You got it. Thank you, Emmett.